Alright, still don't have a phone stand. It's supposed to be here by tomorrow, so it's going to be just a lot of adjusting. We're starting with microbiology today. History of micro is the first section. Historical background. Lazzaro Spallanzani, first to provide evidence to refute spontaneous generation of life. Girolamo Fracastoro, transferable spores causing disease like syphilis. Hooke was the first to see cells in thin slices of cork using a crude microscope. Leeuwenhoek, probably German, maybe. Inventor of the microscope. Semmelweis, hand washing after the in the 19th century, laboring, mother's care. Lister introduced surgical asepsis using carbolic acid. I don't think I knew about the carbolic acid before. Jenner vaccinated a child using cowpox from a milkmaid to prevent smallpox. Koch, father of microbiology, very important, who identified bacillus that caused anthrax. He developed 4 microbiological principles known as Cox Postulates. Causative agent must be found in all patients with disease. You must be able to prepare a pure culture of the agent. The injected agent must cause the same disease in the previously healthy people, and you must recover the same organism from the newly infected patients. Those are Cox Postulates. Graham developed a 7-step process for staining with bacteria specifically. Crystal violet is the blue stain for 1 minute. Wash off with water. Add iodine as a mordant. Wash off with water. Acetone alcohol. Decolorizer removes the blue. Counterstain with safranin, which is red. Wash off with water. So it's a water wash between each step. Gram positive will be blue, and gram negative will be red. Red negative, blue positive. All right, Louis Pasteur, inventor of pasteurization, germ theory of disease, developed a vaccine against rabies and anthrax. Good job, Pasteur. Ehrlich developed chemicals that killed microorganisms like syphilis. There's not a lot to fill in there. Sulfa drugs and salvarsan. I don't know what salvarsan is. Fleming in 1928 discovered penicillin. Ivanowski in 1889 first postulates the existence of viruses while working with infected tobacco plants. Ruska in 1931 first electron microscope to prove viruses existed. Griffith discovered transformation of non-virulent bacteria to a more virulent form. Non-virulent to more virulent. Walter Reed proved mosquitoes carried yellow fever. Salk in 1954 developed the vaccine from killed polio organisms. Sabin in 1955 developed the vaccine from attenuated live polio. Sabin your life, that's funny. Prusiner in 1982 coined the term prion for diseases caused by protein particles. Prion diseases like mad cow disease and Creutzfeldt-Jakob disease. Marshall in 1984 proved that the peptic ulcer is caused by Helicobacter pylori. Page turn. All right, moving on to general principles. You still see that? That's better. Classification of microorganisms. They can be classified by size, presence or absence of nucleus, the nature of the genetic material, the number of chromosomes, nature of the cell wall, and type of cell membrane sterols. Let's take a look at this chart. It's pretty hefty comparisons of microorganisms. Am I gonna go through this right now? Okay, viruses are acellular, have no nucleus, DNA or RNA. The chromosome is nucleocapsid, ribosome none, cell wall none, no membrane sterols. Hopefully I'm reading this right. Okay, bacteria are prokaryotic, no nucleus, both DNA and RNA, one chromosome. I should know what 70S ribosome means, so I will look that up later. Cell wall has peptidoglycan and membrane sterols, none. Fungi, eukaryotic, does have a nucleus, both DNA and RNA, more than one chromosome, 80S ribosome. Well, it's annoying that I don't know what that is yet. The cell wall has chitin, and the membrane sterol is ergosterol. Parasites are eukaryotic. Oops, let's keep this whole thing in view. Parasites are eukaryotic. The nucleus is present, both DNA and RNA, more than one chromosome, 80S ribosome, no cell wall, and the membrane sterol is sterile. Okay, so I have some studying to do in that section. Get some clarity. Not all microorganisms affect humans. Human gut is filled with all kinds, which attribute to the health and well-being of the body. Some of these escape their natural environment. They may cause problems. Whether they cause problems in humans or not depends on the ineffectivity of the microorganism. Ineffectivity is defined as the ability of the pathogen to establish an infection. The infectivity of a microorganism is dependent on its pathogenicity, adherence, invasiveness, antigenic switching, and virulence. Pathogenicity, ability to cause disease, adherence to host cells, like Escherichia, I've always just said E. coli, coli, and Neisseria gonorrhoeae use pili to adhere. Other groups, like Group A strep, and have Fimbriae, which have the same effect. Other bacteria have surface particles that allow them to stick. In each of the above situations, adherence enhances the infectivity of the microorganism by preventing it from being carried away by mucus or being washed away by fluid flow in the bowel or the bladder, thus allowing the organism to colonize the site. Invasion. Ability to penetrate the host defense. Some microorganisms have enzymes that facilitate their invasiveness. Collagenase, C. perfringens, and hyaluronidase, not very good at pronouncing that, assist with invasion. Alright, antigenic switching. Some microorganisms have developed the ability to change the shape of their surface antigens to escape detection by the body's antibody surveillance. For example, trypanosoma, on entering the body, stimulates the production of antibodies for specific antigens found in the surface of the organism. Within a matter of hours, some of the trypanosomes switch the variable surface glycoprotein coat, cover themselves with the new antigenically different VSG coat, slip through the body's defense mechanism, much like the cloaking mechanism popularized by the Star Wars movies. Virulence. Cause serious illness. Some microorganisms produce poisons or toxins, which are extremely harmful to the human. For example, the toxin produced by Clostridium botulinum. Alright, that's a 10-minute video.